Hello students, welcome back to my biology online classes. Today I am going to discuss with you about tyrosine kinases activated receptors, and these tyrosine kinases activated receptors are mainly cytokine receptors and immunology related receptors. Okay, rather than possessing intrinsic enzymatic activity, many enzyme linked receptors act by stimulating intracellular protein tyrosine kinases, with which they are non-covalently attached. Enzyme linked receptor can stimulate intracellular protein tyrosine kinases. Okay, and particularly this family of receptors includes the receptor for most cytokine, example interleukin and erythropoietin, and some polypeptide hormones like growth hormones, whose cytosolic domains are closely associated with a member of family of cytosolic protein tyrosine kinases, the JAK kinases. The JAK-STAT pathway is in your syllabus and very much important. Like Rtk, all of we know about Rtk. The cytokine receptors contain the N-terminal extracellular ligand binding domain, single transmembrane alpha helices, C-terminal cytosolic domain. However, the cytosolic domain of the cytokine receptor are devoid of any known catalytic activity. They have no catalytic activity in case of cytokine receptor. Instead, the cytokine receptor function in association with non-receptor cytosolic protein tyrosine kinase. Functions in association with non-receptor cytosolic protein tyrosine kinases, which are activated, and this is activated as a result of ligand binding. The first step, signal form this cytokine receptor. First step, signal forms cytokine receptor. Dimerization will be ligand induced, and then cross phosphorylation associated non-receptor protein tyrosine kinases. So these are steps. First step is signal from cytokine receptor, then dimerization induced by ligand binding, then cross phosphorylation between associated non-receptor protein tyrosine kinases. Okay, these activated kinase, now this one is being activated. This activated kinase then phosphorylates the receptor. It then phosphorylate the receptor. So this is about cytokine receptor. Let's summarize. There are tyrosine, these are tyrosine kinase activated receptor, and there are three regions: N-terminal extracellular ligand binding domain, single transmembrane alpha helices, and C-terminal cytosolic domain. And with this C-terminal cytosolic domain, it's remain bound with non-receptor. The first step is signal received from external environment, and after receiving signal, dimerization occurs to ligand induction, and then after dimerization, cross phosphorylation associated with non-receptor protein tyrosine kinases. Then this protein tyrosine kinases phosphorylate receptor.